Hi everyone, so in this video I'm going to talk about gene expression or gene regulation in prokaryotes. Um, Gene expression, it's changing the expression of the gene according to the environment surrounding the living organism. So, for example, we know that prokaryotes are polycistronic, meaning that it could code for more than one gene in each mRNA. So this is mRNA. It could code for more than one gene, like this example right here. So an example of this is the lac operon, the one that you're seeing right now. For this example, it's in the in bacteria, and the main purpose of this gene is to break down lactose into glucose and galactose, and both of these are used as energy source. So in bacteria, it's the glucose is actually preferred. So whenever there is glucose, this gene won't work. So the gene will not work if glucose is there. So the mechanism for this, glucose would actually hinder the ability of adenylyl cyclase to work and convert ATP to cyclic AMP. So this process won't occur if glucose is there. Okay, if glucose is not there, then it will just happen normally. So the main reason for cyclic AMP, we need the cyclic AMP as a binding component with cap, and cap is this one in blue, and it goes to bind in this region here in blue, and this is the cap binding site. Okay, this site acts as an activator in the downstream of the promoter. Okay, so this is the cap site to bind, and the cap won't go and bind unless camp, cyclic AMP, is attached to it to make a complex, and then it could actually go and bind. Okay, so this is the first initiating process that could occur. Another one is the repressor, which is this one in the red. This region called the LAC-I, or the inhibitor, and it codes for a protein called allolactose. So in this part, as long as this side is free, there is nothing here could bind there, then it would just go and bind to the operator in this part. This is the operator, and it will just and this is the promoter, and this is the RNA polymerase. So RNA polymerase normally would just go and bind to the promoter and keep working. But if there is this protein binds here in the operator, then it will just block the RNA to just move from transcribing. So it won't be able to move if the allolactose is free like that. So in order for this gene to work, there has to be lactose, because remember that the main purpose of this gene is to turn lactose into glucose and galactose. So if there is no lactose, then there is no need for this gene to be transcribed. So if there is a lactose, it will just go here and bind to the allolactose site, and the allolactose site right now is happy. There is nothing to bind anymore, so it would just stay there, and it won't go and bind to the operator. making the RNA polymerase to move freely. Okay, and then we have different scenarios. So whenever you're having high glucose here and no lactose, then what is going to happen? So as we said, glucose is always preferred. So and there would be no lactose. So what's going to happen if there is no lactose? If there is no lactose, then there is nothing here. So it's free. So it will just go and bind to the operator's site, and it will just hinder the ability of RNA polymerase to just move freely. So the gene is not gonna be expressed. So no expression. Okay. So what if you have low glucose and no lactose? So here, despite the fact there is low glucose, so low energy levels, this gene is not going to be expressed also because there is no lactose. The main reason of this gene is to break down lactose into galactose and glucose. So there is no reason for the gene to be expressed. So no expression here either. And then the third situation, we're going to have high glucose, but this time lactose is available. Okay, so despite the fact we have high glucose, but we also have lactose, so there will be a very low expression here of the gene because we already have the energy, which is glucose, and it's also preferred. So this in this case, it will be low expression of the gene. And then lastly, we have low glucose, and this time we have lactose. And this, of course, there will be expression because we have low energy and we have lactose. So it will be broken down into extra glucose and galactose. So in this case, there will be expression. And that's it. Thank you for watching.